All hope you all are doing well. Today we are going to discuss the estimation of blood glucose by glucose oxidase peroxidase method on the colorimeter. This is the second video on my video series of practical biochemistry, and it is one of the core topic of the practical aspects related to the biochemistry. It is frequently asked in a practical exam. So in today's video, we will discuss what are the various different methods available for the blood glucose estimation. Then we will see estimation of blood glucose by GOD-POD method in the detail, along with the various VIVA questions asked frequently in the VIVA from this particular topic. And in the end, we will see hands-on estimation of blood glucose by GOD-POD method on the colorimeter. So please watch this video till the end, and before going in the detail, please subscribe my YouTube channel, that is Biochemistry Basics by Dr. Amit, and don't forget to press the bell icon so you can get all the notifications from it. So aim is to estimate blood glucose in the given sample. And the introduction is the term blood sugars include all the hexoses, even though main hexose present in the blood is glucose. And the blood glucose level is an important indicator of the body function, and it is actually maintained in the normal range with various hormones. Two main hormones which are regulating the blood glucose level is insulin, which is a hypoglycemic hormone, and glucagon, which is a hyperglycemic hormone. If there is any abnormality of the blood glucose level, it indicates disturbances of the carbohydrate metabolism. And the one important lifestyle disorder associated with the abnormality of the blood glucose level is diabetes mellitus, which is characterized by hyperglycemia. That means increase in the blood glucose level, which occurs due to the absolute or relative insulin deficiency. Now, methods for the glucose estimation. So there are the various methods available for the glucose estimation. Among this, three methods are important one. First one is the reduction method, second one is the condensation method, and the third one is the enzymatic method. In the reduction methods, examples are Follin-Wu method and Nelson-Somogyi method. In the condensation method, example is the orthotoluidine method, and in the enzymatic method, examples are hexokinase method and glucose oxidase peroxidase method. Among this reduction method and enzymatic method is more specific as compared to the reduction method because there are very few interfering substances are there and the reagents are more stable as compared to the reduction method. And this enzymatic methods, they measure the true glucose. Fine. So these are the advantages of enzymatic method. While in the reduction method, estimation of glucose by the reduction method, there will be the various interfering substances will be there. Various interference by the various reducing substances like uric acid, bilirubin, vitamin C. So it will not measure the true glucose value. So that is the one of the disadvantage of the reduction method. Another disadvantage of the reduction method, it is a multi-step approach. Fine. While the enzymatic method, there is a single step. Only. Another disadvantage of reduction method is it doesn't measure the true glucose value. The only advantage of reduction method is they are cost effective as compared to the enzymatic method, but nowadays, the frequently used method for the glucose estimation is enzymatic method, and in the enzymatic method, examples are hexokinase method and GOD-POD method. In this video, we will also discuss the principle of hexokinase method, and we will also discuss why hexokinase method is better as compared to the GOD-POD method. Estimation of blood glucose by glucose oxidase and peroxidase method. The principle of glucose estimation by GOD-POD method is glucose present in the blood is converted to gluconic acid and hydrogen peroxide by glucose oxidase, and the hydrogen peroxide is catalyzed by the peroxidase enzyme to the H2O and nascent oxygen. This 4-amino antipyrine, which is an oxygen acceptor, will accept this nascent oxygen, and in the presence of phenol, it will be converted to the pink color chromosome. And the intensity of this pink color will be directly proportional to the concentration of the glucose present in the solution. If there is a darker pink color, that means glucose present in the blood or in the solution is higher. And we have to measure the intensity of the color at 520 nanometer wavelength, and we have to compare it with that of standard treated similarly. So that is the principle of glucose oxidase method, glucose oxidase peroxidase method, which is asked frequently in the exam. Now reagents required for the glucose estimation by GOD-POD method. So the first one is the glucose reagent. In the glucose reagent, it is consists of glucose oxidase, peroxidase, 4-amino antipyrine, and phenol. Along with the glucose reagent, we also require glucose standard, and the value of the glucose standard is non, which is 100 milligram per deciliter. Now, procedure for the glucose estimation by GOD-POD method. So first you have to take the three test tube and label them as blank, standard, and test, and then proceed as follow. So first take the blank in the blank test tube, that is 10 microliter. Then take the standard glucose standard in the standard test tube, 10 microliter, and then add unknown sample 10 microliter in the test tube. Fine. After taking the blank, standard, and test, add 1 ml of glucose oxidase peroxidase reagent in all these three test tube. Mix thoroughly and incubate at 37 centigrade for 10 minutes. And after 10 minutes, you have to read the optical density on the colorimeter at 520 nanometer or gain filter. After taking the optical density, you have to feed the optical density in this formula, that is optical density of test minus optical density of blank divided by optical density of standard minus optical density of blank multiplied by concentration of standard. Concentration of standard is non, which is 100 milligram per deciliter. After feeding the optical density in this formula, you will get the blood glucose. Now, normal range of blood glucose. So there are the various normal ranges of the blood glucose are there. First one is the fasting blood glucose. The fasting blood glucose, the sample is collected after 10 to 12 hours after fasting. Then there is a postprandial blood glucose, PP2BS. The blood sample is collected after 2 hours taking the meal, and the random blood glucose. In the random blood glucose, blood sample is collected randomly. So the normal value for the fasting blood glucose is 70 to 100 milligram per deciliter. For the postprandial blood sugar, it is less than or equal to 140 milligram per deciliter. And for the random blood sugar, it is 80 to 140 milligram per deciliter. So these are the various normal ranges of the blood glucose. You have to remember this normal ranges. It is frequently asked in a VIVA as well as in the theoretical exam. Now, interpretations of value. So first we will see the hyperglycemia, the reasons, available reasons for the hyperglycemia. So there are the physiological reasons and there are the pathological reasons. So the physiological reasons for the hyperglycemia are after high carbohydrate diet, emotional, that is stress, anger and anxiety, that also can lead to the hyperglycemia, and pregnancy. So pregnancy can also lead to the hyperglycemia. And the pathological reasons for the hyperglycemia is insulin deficiency, that can be absolute and relative, which leads to the diabetes mellitus. If there is any hyperactivity in the adrenal gland, thyroid gland, and pituitary gland, then they can also lead to the hyperglycemia. These are the various physiological and pathological reasons of the hyperglycemia, and the most common one is the insulin deficiency, which leads to the diabetes mellitus. Now, common causes of hypoglycemia, that means reduced blood glucose concentration. The physiological are during starvation, after severe exercise. So these are the physiological reasons for the hypoglycemia. And the pathological reasons for the hypoglycemia is overdose of the insulinoma, and if there is a decreased activity of the pituitary gland, thyroid gland, and adrenal gland. Along with that, glycogen storage diseases. This all can lead to the hypoglycemia. And remember, hypoglycemia is more dangerous as compared to the hyperglycemia. If the blood glucose level is less than 45 milligram per deciliter, then it is a critical condition. These are the various reasons for the hypoglycemia. Now let's see the VIVA question. The first question is explain principle of the hexokinase method. The hexokinase method principle is hexokinase convert glucose into the glucose 6-phosphate, and this glucose 6-phosphate is oxidized to the 6-phosphogluconate by the glucose 6-phosphate dehydrogenase, which leads to the synthesis of NADH. And we have to measure the absorbance of this NADH at 340 nanometer wavelength. This hexokinase method is better as compared to the glucose oxidase peroxidase method because there are the very few interfering substances are available in the estimation of glucose by the hexokinase method, and it is a reagent is more stable, and the linearity of this method is good as compared to the GOD-POD method. That is the principle of hexokinase method. Now, what are the fasting, postprandial, and random blood glucose samples, and what are their normal glucose levels? We have already discussed the normal values of the fasting, postprandial, and random blood glucose levels. Third question is why fluoride vacuoles are used for the glucose estimation? Because in the fluoride vacuole, there is a presence of sodium fluoride which inhibits the enolase enzyme of the glycolysis. That's why they are used for the glucose estimation. Explain the principle of plasma glucose estimation by GOD-POD method. We have already discussed in the earlier part of this video. What are the causes of hyperglycemia? We have already discussed that physiological and pathological. In the pathological, hyperactivity of the thyroid, adrenal, pituitary gland, and diabetes mellitus. What are the causes of hypoglycemia? Again, we have already discussed that the physiological and pathological. In the pathological part, it is due to the overdose of insulin, insulinoma, and the decreased activity of the pituitary gland, adrenal gland, and thyroid gland. What are the other methods used for the glucose estimation? We have already discussed in the beginning where there are the various reduction methods, condensation method, and enzymatic method. In that, enzymatic method is more specific. What is HbA1c? HbA1c is the glycated hemoglobin which is formed by the non-enzymatic reaction. The glucose unit will be attaching to the globin chain of the hemoglobin non-enzymatically, and we can get the average glucose value for 120 days. So that is the glycated hemoglobin, and this glycated hemoglobin is the advanced test available for the diagnosis as well as for the monitoring of the diabetes mellitus. What are the tests for the diagnosis or diagnosing diabetes mellitus? So the first test we have already discussed, that is glycated hemoglobin. If the glycated hemoglobin is more than 6.5%, then that is one of the criteria for the diagnosing diabetes mellitus. Another test we can do for the diagnosis of diabetes mellitus is the oral glucose tolerance test, which is called as a OGTT. Another question is what is the diagnostic criteria for the diabetes mellitus? So the diagnostic criteria for the diabetes mellitus laid down by the American Diabetic Association is if the RBS random blood sugar value is greater than or equal to 200 milligram per deciliter with classical symptoms of diabetes mellitus, that is polyuria, polyphagia, and polydipsia. If the fasting blood sugar value is greater than or equal to 126 milligram per deciliter, and the 2-hour post-load glucose value in the OGTT is greater than or equal to 200 milligram per deciliter, and if the glycated hemoglobin is greater than 6.5%. So these are the criteria laid down by American Diabetic Association for the diagnosis of diabetes mellitus, and everyone should remember this by heart. Now, how blood glucose is regulated? So, blood glucose is regulated by the various hormones like insulin and glucagon. Insulin is called as a hypoglycemic hormone, and glucagon is called as a hyperglycemic hormone. So, blood glucose is regulated with the help of these two hormones. Now, let's see the demonstration of blood glucose estimation by glucose oxidase and peroxidase method.
So as we have already seen the theoretical part of estimation of plasma glucose, now we will see how to do the operational procedure for the plasma glucose. So for the estimation of plasma glucose, various requirements are: first, there are the 3 test tubes, then you will also be needing various micropipettes along with the tips. So these are the volumetric pipettes, fine. So this both are the volumetric pipettes, and these are the macro tips, and these are the micro tips. Now you have to see how to insert this tips into the tip holder of the pipettes. So this is the way you have to hold the tip into the tip holder. Fine. So now we will see first step for the estimation of plasma glucose is labeling of the test tube. Fine. So the first you have to label the test tube. First is the blank. Then you have to label standard. Second test tube, label it as a standard. And the third test tube, label it as a test. After the labeling part is over, now you have to take the 10 microliter of blank into the blank test tube. So in the blank we are taking the distilled water, so take the 10 microliter of it and add that 10 microliter of blank in the test tube. Make sure that it is in the bottom of the test tube. Then you have to take the 10 microliter of the standard. The concentration of the standard is it is 100 milligram per deciliter. So take the 10 microliter of the standard in the standard test tube. Fine, so this is the standard test tube. And then you have to take the 10 microliter of the test. The concentration of the test is unknown, we have to find it out. So take the 10 microliter of the 10 microliter of the test in the test tube. After the adding blank, standard and test, now you have to add 1000 microliter of GOD-POD reagent in all these 3 test tubes. So you have to take the 1000 microliter of the GOD-POD reagent and first you have to add into the blank test tube. So this is the blank. After adding the reagent, mix it properly. Then again take 1000 microliter of the GOD-POD reagent. Add into the standard test tube and mix it properly. And again take the 1000 microliter of the GOD-POD reagent and mix it properly in the test tube. After mixing it, you have to keep all these 3 test tubes into the incubator for the 10 minutes. So this is the incubator. The temperature of the incubator is set at 37 degree. So you have to keep it for the 10 minutes. So this is after 10 minutes of incubation period. As you can see, there is a color change in the standard and there is a color change in the test. As the concentration is higher, the color will be the more darker. So as you can see in the test, color is more darker as compared to the standard. Now we will take the optical density in the colorimeter. We have already set the 0 by giving water blank in the colorimeter. So now we will take the optical density of the blank, standard and test. So you have to take that blank in flat bottom test tube, transfer that reagent in the flat bottom test tube and take the optical density. So the optical density of the blank is already taken. Then take the optical density of the standard. So this is the optical density of the standard, 0.34. And then take the optical density of the test. So this is the optical density of the test. Now after taking the optical density of the blank, standard and test, you have to feed that optical density into the formula and then you have to calculate the concentration. So this is how you can do the estimation of glucose with the help of colorimetry by GOD-POD method. Thank you for watching. Thank you.